Miron. And yeah, thanks a lot also for challenging me from time to time with your questions. It's always nice to, think about other things than answering emails and doing day-to-day work. So that's why it's also actually a pleasure for me to talk about my favorite topic to you today, which is RNA, small RNA, microRNA, long RNA biomarker discovery, which, as you said, is something we've done for quite some time here in Tamerna. Okay.
Sorry, we cannot hear you. It seems that maybe you mute yourself or.
Yeah, it seems I muted myself. Yeah, should work now again.
Yeah, it's okay.
Yeah, so let's see. Hopefully, yeah, should be fine now. Okay, yeah. So more than 10 years ago, we launched Tamerna. We are located in Vienna in so-called Vienna Biocenter. It's quite nice ecosystem of companies and academic institutions. And what we're trying to attempt for the last 10 years is to identify and discover microRNA biomarkers, and then based on that knowledge that they are biomarkers, to develop diagnostic tests with ultimately the aim that these tests will provide decision support. So for me, the buzzword when I talk about biomarkers is really decision support. It means you measure something, and by measuring that you can generate information that will have an impact on decisions. And we're looking at applications for that in the molecular diagnostic space. That means providing decision support to clinicians, to facilitate how they manage their patients, but also drug development, because as you may know, biotechs, pharmaceutical companies, when they go through all of the stages of drug development, there are a lot of questions they have. And some of these questions we can also try to answer with biomarkers, especially microRNA biomarkers. For instance, questions related to the safety of a drug or the efficacy of a drug. So that's what we're trying to do. And we've been set up for, I would say, the first 7 years really as more or less as a sort of an academic lab outside academia. So we've did a lot of research, a lot of development. And ultimately, in one project, which is on liver function and using microRNAs to assess liver function in patients, we've been quite successful, and that led to a lot of changes. So we had to then really set Tamerna up as a manufacturer of in vitro diagnostic tests. This is what we accomplished. So we implemented something very unusual for academics, a quality management system. We did this according to the ISO 13485 standard, and are now sort of also, I think, one of Europe's first accredited manufacturer of a microRNA-based CE-IVD test, which is actually already used in a couple of hospitals in Austria, Germany, and Switzerland. Yeah, and beside now that more commercial life, we're still very much interested in research and R&D. That's why we're also happy to join projects such as Atheronet, previously also CardioRNA, and a part of CardioLink. So we try to network, we try to exchange knowledge, and yeah, that's why I'm also here today to share a little bit of our knowledge. Yeah, I mentioned this just to start really from the basics. Biological markers, so as I said on the first slide, it's something we can measure. It can be anything, molecule, a gene, whatever. And by measuring it, we can generate information about pathology or physiology. And somehow, hopefully, we can then make use of that information to make better decisions. And just to give you a few examples, when we talk then about more a diagnostic test, it always has an intended use with it. And the intended use describes exactly how that biomarker should be applied. And this can be as much as diagnosis of a disease, but there are also other use cases, prognosing of a disease outcome. That's what our test does that we've developed. We're trying to predict the risk of liver failure as an adverse outcome of liver surgery. You can also try to predict treatment response. This is more than in the direction of a companion diagnostic. It means a diagnostic test that must be performed prior to a drug being prescribed to a patient, and so on. So there are many different use cases that can be developed, and we do think that for some of those, microRNAs or RNAs in general can be quite good biomarkers. And before I go into more research topics, I want to give you another overview of what it means to actually, go from an idea or a planning phase to a product. And usually this, or in our experience, I'm not going into details here of this table. Essentially, there are two phases in that. The first phase is to discover and validate biomarkers, and that ends approximately here after biomarker discovery, maybe some verification. That means you have identified RNAs that are tightly associated with a clinical outcome. And once you have validated and you've defined that association, the next stage, and which is less research, it's really more development. It's developing the diagnostic test that then ultimately quantifies the biomarker and provides an actionable result. And what I'm trying to do today is just to highlight a few concepts, especially in relation to microRNAs and other types of RNAs, how to best perform biomarker discovery. So not going into these details, the very technical details of developing a diagnostic test and what that means, but really what may we have to consider while we are seeking or while we are hunting for new RNA biomarkers. So that would be the main topic. And something that's important to understand when we're doing biomarker discovery is that we have to take very important decisions at a very early stage, actually. What do I mean by this? So here you can see just the stages that we usually go through. We have a discovery, and then we go with our certain biomarkers into validation. And at some point in this whole process, we have to make a decision. We have to make a design freeze, and we have to say that's our panel of microRNAs or RNAs that we're going to work with, and for who we are then developing a diagnostic test. And sometimes we even take that, correctly speaking, during validation or prior to validation. So what it means, you have to take that decision on what are your targets potentially quite early on when you have measured a large number of biomarkers, but in a relatively small sample size. And what's happening afterwards, you do a prototype development and you perform large clinical studies. And that means that with increasing sample size, the variability increases. So this is the ISO development. This is where you have a prototype. And ultimately, at the very end of the day, you will generate data that matters. But you'll generate data which matters for a selection of biomarkers that you have done very early on, maybe on a limited number of samples. And it can cause some problems, because it means that you have to pick the quote-unquote right biomarker candidates early. And what are the right biomarker candidates? It's the ones where you can really optimize signal or have the highest possible effect size due to your disease of interest, the clinical outcome, whatever, compared to the noise. So we have to make sure that we look for the right signal, and at the same time understand what are the causes of noise in the data, of variability, something where our signal has to stand out of, and try to minimize this as well. And then this is all about, when it comes for me for biomarker research, we always have to look for high signal to noise. Now, applying this to cell-free RNA and cell-free RNA biomarker discovery, they're obviously, the signal is clear. Our signal should be a circulating cell-free microRNA or extracellular vesically associated microRNA or longer RNA. That's clear, but there is a lot of potential sources of noise. And this was actually nicely reviewed by also Miron here and David and Ivan not so long ago in the context of long non-coding RNAs, but this in the same way applies also to small RNAs. So I'm not going into too much detail here, because this is really well summarized, and just maybe to highlight that clearly, how you collect the sample plays a major role and is a source of noise. What the sample type you're working with, what are potential impacts on sample quality. Hemolysis is a common factor that can introduce variability simply by the release of RNA from destroyed red blood cells. Certain, some biological effects, and then the whole assay. So there's a lot of things that can come together that increase noise, and ideally you understand those sources very well. And luckily, in the context of cell-free RNA, I think especially cell-free microRNA, that's why I'm referring you to that review. I think we understand quite well. So I'm not going into detail today to explain again differences of plasma and serum and the importance of hemolysis and sample processing. That's, I think, quite well understood. I'd like to focus on a few other topics that maybe just give you an additional perspective on the choices you can make when seeking an RNA or a microRNA biomarker. So I'd like today to talk about briefly a topic that's very close to my heart. I've been working on this for some time, and it's how do we normalize small RNA or microRNA sequencing data? What could be best practices there and things to consider? I'd like to introduce a topic that was not so known to me until recently, and I'd like to share this on biologically active versus inactive microRNAs, and how we may be able to utilize and leverage that for biomarker development. Extracellular vesicles, definitely a hot topic, carrier of different molecules, including RNAs. So should we look at extracellular vesicles? Yes, no, and why, for instance. And then finally, I'd like to close with a look beyond small RNAs, so long cell-free RNA, and how we, at least internally, have now attempted to set up next-generation sequencing workflows to do a full genome-wide analysis of such longer cell-free RNAs, such as cell-free mRNA, but also cell-free longer non-coding RNAs, etc. So that would be my topics for today. Let's start with the normalization. So for those of you who had the opportunity to look at small RNA sequencing, microRNA sequencing data, they probably have heard that the standard normalization that you apply to visualize your data is so-called reads per million. It also is done for general RNA-seq, then you also adjust for the size of or the length of your transcript, and it's read per kilobase, or also transcripts per million, but it always has this per million. And the per million implies that your total, the total amount, or that of RNA in that sample actually remains constant across the experimental groups. So it assumes that between your experimental groups, there's no difference in the total amount of your RNA of interest. Because you say, I always scale or normalize per million. That million is a constant. You just account for sequencing data. However, it's been found already in 2017 by Michael Nodine in his group that there are certain, cases where this assumption is not true. One is, for instance, plant development. So as indicated here, during plant development stages, you can see that the total amount of microRNA per microgram of plant mass actually increases. So you don't have anymore the assumption when you then would like to compare transcription between these states, the assumption that it's actually you have always a 1 million constant reads simply does not apply. And that's why actually originally they had developed the concept of using spike genes, which I'll come to. Just another, maybe more extreme example. So we are still interested in microRNAs that are truly enriched in cerebrospinal fluid or brain, and which I'll show you a bit of data later on, which have actually higher concentration in CSF compared to serum. So we wanted something that's super abundant in CSF, low abundant in serum. So if there is brain damage or CNS damage, that could leak and result in a measurable increase in serum. So we did a lot of sequencing to compare CSF and serum. And what we noticed is that the RNA composition is completely different. So you can see here color-coded the different types of RNAs found in these biofluids. And it's quite obvious this microRNAs are, overall, there's much more microRNA in serum compared to CSF in terms of, and that can, it's again, that when you try to compare and want to find something that's truly more abundant in CSF, that RPM is a bit challenging. So that's why we adopted that strategy published by Michael Nodine in 2017, who had designed these spike genes in the context of plants and applied this, so that ultimately the idea was that we are not anymore dependent for the normalization on all the other RNAs, because reads per million depends, this in red is your target microRNA, but you need all the other RNAs and the copy numbers to normalize those. So you're dependent on the RNA composition. And the concept of spike genes is that you add a constant defined amount of these artificial oligonucleotides to each sample. And then rather than using all the other RNAs or microRNAs in your sample, which are now shaded in gray, you only use the spiking calibrator to standardize. And so you become independent in your normalization, and you can go towards an absolute quantification, actually. So just briefly, what we call those now, we call them the MIND spike genes. So they have been designed to be usable for pretty much any mammalian species to mitigate bias from ligation, and some may be over underrepresented. They have randomized 5' and 3' ends. This results in sort of a really unbiased representation in your sequencing library, independent of which library prep method you're using, and they only have a defined core sequence that differs between the individual spike-ins. And they have a molar concentration that we have optimized so that they're pretty broadly usable from cells, tissues to extracellular vesicles, because more or less we've seen that microRNA concentration ranges in these molar amounts. Yeah, so the workflow is easy. You have your small RNA sequencing protocol, and then to the total RNA, to the defined volume amount of total RNA, we spike in these, and then we recover the spike-ins from the data. It looks like this, that for every sample in an experiment that you have sequenced, we derive the count of each of these 7 spike-ins, and we plot it against its molar concentration, and we can derive, first of all, what is also nice, a quality control parameter for that run. But then further on, we can also do a regression analysis. And what you can see here on the right is then that we can overlay the concentration of these of the spike-ins with all of the detected RNAs. And then to make this more visible, what this allows you then between 2 samples, here, for instance, you have, I think this was plasma, and this is from extracellular vesicles. You can see that while in plasma, you have a fairly large amount of microRNAs in this high abundance, so above spike-in 3, 2, and even 1. You can see 1 is up here. You can see that in this sample, you have a much lower, first of all, complexity, but also overall abundance, as most of the microRNAs scatter between spike-in 6 and 5, while here you have much more. And this is something that you don't see with RPM, but this is something that the spike-ins then allow you to see, and that's something we like to leverage. So coming back to the comparison of CSF, cerebrospinal fluid, and serum. So we analyzed our data in 2 ways. This is, again, the RPM, where we would assume a constant amount of microRNAs in both serum and CSF. So we would scale to this artificial 1 million reads. And you could see if we do this scaling, then it looks like we have a fairly large cluster of microRNAs that give a higher signal. So, brighter color here in the CSF. But when we replot the heat map now with the absolute concentrations that we have derived from the spike-ins, we see that when you look here now, it's a bit tricky. CSF is now left, serum is right. But when you look at the color code, you can see for most clusters of RNAs, we have brighter color in serum. And there's actually only a very small cluster now of microRNAs where we truly have, on an absolute level, a higher concentration. And these are now clusters of CNS or brain-enriched microRNAs that we're following up in an EU project called Transpireline as potential indicators of neurological damage. But we found this extremely helpful and useful to pinpoint us to the right biomarkers. So just to show you that these spike-ins, we can actually use them really across a broad range of sample types. So each data point is actually the median of the percentage that the mind spike-in reads took up of total reads, because we were initially worried that we may overload or flood our library with these spike-ins. So we wanted to also choose a concentration that would only take up a minimal percentage. And you can see this is 1% of total reads. And we can see most projects that we do that on average have 20 to 50 samples. We can see that the median percentage that the mind spike-ins take up of the total sequencing depth is quite low. It's below 1%. And that works well in biofluids, tissue cells, EVs, and also looking, for instance, at just protein fractions. And we now have applied this to, I think, sequencing more than 1,700 different biofluid samples with this workflow. And we can really understand now quite well the complexity. You can see here is plasma and serum, in our experience, have the highest complexity of microRNAs. We detect routinely more than 1,000, while we have seen, in feces or CSF, and we have quite lower complexity. And if you want to sometimes compare across them, spike-ins are really useful. So with this part, and related to the discovery of small RNA and specifically microRNA biomarkers, for us it was really a big improvement to implement these controls, because it mitigates the risk that we have biased results due to changes in the total RNA composition. If you're interested in taking this up in your own small RNA sequencing workflow, please just shoot me an email. We're happy to help you and to work with you on this. Okay, coming to the next part on biologically active versus inactive microRNAs. So what does this mean? It means that when you look at the localization of microRNAs inside a cell, clearly the microRNA, after its maturation, it's incorporated, it's bound by agoproteins, and there's dicer, it's cutting off that loop. And then what happens then is the RNA-induced silencing complex that consists of ago, the microRNA, but a range of other proteins, also potentially the ribosome if this interaction mRNA is formed, so the risk. That means that we can find agoproteins and RNA in these two different sites inside a cell. So either here before loading into the RISC or in the RISC. That this is actually the case has been shown already in 2015 in this PNAS paper by La Rocca et al. And what quite fascinates me is that they found in normal tissue, when they stain for these AGO2 proteins using in a Western blot, you can see that in most tissues, heart, muscle, lung, you have most AGO actually present in this low molecular weight. So it's not in the RISC, and it's actually in a state or in a form where it hasn't bound to mRNA, where it's actually not active. There's only a few tissues or cell types, kidney or brain, where they did find a considerable amount of AGO2 proteins inside the high molecular weight complex. So, exerting its function. When you go to cell lines, that are actively proliferating, you can see that this is a bit different. You see much more presence of AGO2 in high molecular weight. Why is this relevant? Because this is not a constant attribute of cells or tissues. It actually depends a bit on their states. So in this example, then in the same paper, what they did, they analyzed this in resting T cells. So with the activating antigen being absent, so also in resting T cells, they observed most of AGO and essential microRNA is these low molecular weight complexes, so presumably inactive. However, when they have stimulated the T cells, they suddenly observed a redistribution of AGO more to high molecular weight. So it means that after exposure of the T cells to their or T cell receptor to the matching antigen, that suddenly activated microRNA regulation. And that was also shown here. So the recruitment of a lot of microRNAs from low to high molecular weight, from resting to stimulated. But what's interesting is that this did not always overlap with also an increase in transcription or expression. And that's seen by a lack of correlation between expression change and redistribution. So it means that without changing the overall level or abundance of microRNAs in the cells, there was a change of activity. And this is actually something that you only see if you start to stratify your analysis or focus your analysis on active or inactive. This is shown here in this correlation plot. So some microRNAs changed in expression, but not in distribution. Some did redistributed highly, but overall the level remained the same. So the question here is, do we need to consider this when we are conducting cell-free microRNA analysis or microRNA biomarker analysis? To do this, ideally we have a method that allows us to quite rapidly do this kind of selection of risk complexes from cleared from a lysate, from a sample. And this is then was published in 2020, a method in nucleic acids research by Grenzinger et al., who developed a procedure called TRAP-R. So it's actually a column-based method where you, from a cell lysate, but potentially also from the lysate of a biological fluid, you can enrich for microRNA of RNA that is inside the RISC complex, and you can deplete RNA or microRNA that is not in these complexes and potentially inactive. And this is more or less also, I don't get paid by them, by Lexogen or anything. They have built a kit around that, the Cropper kit, and we have now used this several times because it's a fairly easy way to do this. So we've done a study where we tried then to better understand what is the impact on our results when we add this step of RISC complex enrichment compared to just the normal total RNA analysis. And we tried to do this on cells on extracellular vesicles from plasma, on urine and plasma. And for each of these, we had 3 replicates where we did, just as we always have done it, total RNA extraction directly from the sample, and then a second number of 3 replicates where we did the risk enrichment prior then to small RNA sequencing using our platform. And this is what we observed, and this is yet unpublished, but we've seen quite a lot of impact of risk enrichment. So what you can see here is the RNA composition of plasma, plasma EVs, urine and cells. You can look at microRNAs, that's always the red bar. You may also look at tRNAs and piRNAs, green and yellow and blue, the ribosomal RNA. And when you can see for plasma, for instance, when we do the risk enrichment, we lose microRNAs, so only a subset of microRNAs really seems to be inside the RNA-induced silencing complex in plasma. We don't lose so much ribosomal RNA, which when thinking about it makes sense that this is already maybe a complexation of the risk with mRNA and ribosomes bound. We haven't seen, and I'll show you, too much effect on plasma EVs, interestingly, although the content, I'll also come to that, of microRNAs, at least to our experience in plasma EVs, is fairly low. In urine, we've seen huge impact. So you can see urine, if we don't do the risk enrichment, we get microRNAs, we get a lot of tRNAs, piRNAs, but if we do the risk, we mainly end up with some microRNAs and again, some ribosomal RNA. And also inside cells, we've seen that in the control, so total RNA extraction, we get some tRNA fragments, etc. And if we do the risk enrichment, we mainly get microRNAs. Yeah. So all in the composition, we've seen it really had a major impact in urine and cells, medium impact on plasma, and relatively little impact on plasma EVs. So now in terms of microRNA levels or profiles themselves, you can see in this heat map, I have to minimize this, that, well, first of all, each matrix clusters, which makes sense. Yeah, I mean, we have plasma EVs, plasma, urine, and cells, each building a cluster. But when you look inside each cluster, you do see that here we always have a subgrouping according to whether it was the risk-associated RNA or the total RNA. And you can see quite clearly, especially for cells, this is a very tight subclustering. So for cells, it made a big difference whether we look exclusively inside the risk or the total RNA. Here we see a strong enrichment of certain microRNAs that seem to be biologically more active. But we did see this subgrouping for all of the matrices that we've looked at to a lower degree. So for urine, for instance, when you look in risk, you're lacking suddenly certain clusters of microRNAs, for instance. You can also express this as a volcano plot, just again to illustrate the impact. And I have to say, we've seen really the strongest impact of risk enrichment in cells, in urine, and then to a lower extent in plasma or plasma EVs, which tells me that, especially in EVs and in plasma, it seems that really most of the microRNA that's secreted is already in the risk complex, while this, again, it may not be true intracellularly or urine. So whether you do or you implement this risk enrichment prior to your analysis will have probably the biggest impact when you're working with tissues or cells as your biomarker source. Potentially also urine, which you may have, depending on how the urine was processed. If it's sediment or centrifuge, you may also have a higher residual presence of cells in there. But in plasma EVs and plasma, I'm not showing plasma here, it had a rather low impact. Yeah, so it really depends. But if you were to hunt maybe for biomarkers using cells and tissues, this could be a worthwhile addition. So to summarize this part is that we observed that intracellularly microRNAs are localized either outside or inside the risk complex, outside the risk complex in an inactive state. This can change. There can be a redistribution depending on, for instance, an external stimuli or disease. And to a certain extent, also in biofluids, we observed that microRNAs are present both inside and outside the risk complex, which depends a bit on the type. And so it's something that I think could be worthwhile to research further because I think this has mainly been neglected so far, and there's very little literature on doing this type of separation. So I think that's quite an exciting area to further understand what the impact of such a risk enrichment could be. Right. Yeah, I think that's it. Okay. So the next part where I'd like to share a few insights that we made over the last years with you is on extracellular vesicles and what their importance or what their impact could be on cell-free RNA, microRNA biomarker development. And that actually coming back here, that actually links a little bit with the last part as well. Yeah, it's a bit of a teaser, but let's stay with small RNAs for now. So cell-free microRNA, as we understand, or as I think about this now, there are essentially two main mechanisms by which cell-free or circulating microRNA actually arrives outside the cell. So there's something that I call active release. So these are mechanisms such as the formation of exosomes from these microvesicular bodies, and then releasing these small particles that could be larger microvesicles or extracellular vesicles that contain RNA. And there is also potentially RNA outside of these vesicles. They are bound to lipoproteins or other types of proteins. And presumably this all has to do with the sort of a regulated or active secretion. And we think that this may correspond to the intracellular changes and may be informative, or these levels may inform about pathology, pathophysiology, inflammation, fibrosis, etc. In addition to that, there's now in our understanding a second mode of action that colleagues, Warren Glaab and his team at Merck actually thought about, which is called passive release. So it's really that in an acute setting can be toxicology. They looked at this really in drug-induced cell or drug-induced organ injury, but also in acute disease, traumatic brain injury. Harsh acute challenges to tissues. You have something called a passive release, which is pretty much due to the plasma membrane becoming leaky and just resulting in leakage of microRNAs into the supernatant, which is yet second mechanism by which microRNAs can end up in biofluids. So the question is, these two mechanisms, how are they reflected more in the extracellular vesicle or in protein complexes, and how overall, more important may it be to distinguish when we measure such cell-free circulating microRNAs to look at different carriers. And there I like to just come back also to a paper published quite a while ago from Jason Arroyo, who did this, in my view, really elegantly. What they developed is, or actually validated, is a size exclusion chromatography protocol where they validated it with the performance of this fractionation and in which fraction they can find certain sizes of proteins, smaller to larger, or extracellular vesicles. You can see just the data here from their supplements. I think this was quite nicely done. And so you can see in around fraction 9, you have really a peak of vesicles with around 100 nanometers diameter, while in later around maybe fraction 16, 15, 16, you have probably the size of ago. Ago is 80 kilodaltons. Yeah. So you have a clear separation, I would say, of Argonaut carried RNA and vesicles. And then what they did back then, still with qPCR, they fractionated plasma pools into these fractions. You can, again, 8 to 9 are these 100 to probably more than 100 nanometer diameter vesicles. And then you have these Argo carried proteins probably around here, fraction 15, 16, 17. And then they back then did the qPCR array, so they did not sequence. They just looked at a targeted panel of microRNAs. And then in this heat map, they plotted the intensity or the signal that they got from these different fractions. And what they've seen is that they're fairly small subset of vesically enriched microRNAs, so where they had predominantly signal in the vesicular fraction. Then there's a certain group of microRNAs that seem to be present in both vesicles as well as protein complexes, and then a fairly large sort of protein enriched. And some microRNA 122 from the liver, it's a liver specific microRNA, is mainly found actually in large, in smaller protein complexes, and there's no detectable presence, at least with their method, in these. So they have seen that, that's why they call it the most, or the majority, is actually because they've seen clearly more microRNAs giving a predominant signal in the protein fraction. However, there are a few caveats to that. So this is shown for 3 microRNAs here, for instance. You can see that LED7A has a peak in the EV fraction and a smaller peak in protein, while MIR16 and MIR92A in plasma show the peak in protein fraction. However, when you look at that in serum, and coming back to what I said initially, matrix is important sources of noise. It's really important to consciously think about what sample you're using. So when you look at this in serum, suddenly you do see a peak of microRNA here, in the EV fraction. So that's something that the serum collection, the tubes, whatever's happening, can give rise also to the maturation of vesicles. So that simply tells, yet again, plasma doesn't equal serum, and we may not be able to reproduce observations from one matrix in the other matrix. So for our own research within Tamerna, we found it quite interesting and relevant, because we have been and we are still interested in platelets, so in thrombocytes, and especially we are interested in how the activation of platelets through different agonists, ADP, thrombin, thromboxane, overlaps or results in the release of microRNAs. And the question is always, are these activated platelets releasing microRNA mainly in EVs or in protein complexes? And can we, for instance, improve sensitivity or specificity for that mechanisms if we focus on one or the other fraction? And that's what motivated us to look into this. So with our collaborators in an Austrian project called BusGauge, and Professor Manuel Meyer and his team, we had the opportunity to analyze a few relevant samples to address this question. So on the one hand, PPP or platelet poor plasma, that was using ultracentrifugation fractionated in small EVs, large EVs, and the EV depleted supernatant, so presumably where you have microRNAs and protein complexes. But what was done at the same time, the platelets here that have been centrifuged to obtain platelet poor plasma that was fractionated were then resuspended in buffer and then activated. And from more or less the same volume corresponding to the original one, then the supernatant here was taken and again fractionated into small EVs, large EVs, and EV depleted proteins. So we could really understand or resting and then activated platelets. And then yet again, we sequenced this, and you've seen this again, RNA composition. Just to come back what I said initially when talking about normalization, what's striking is when you look at the RNA composition here, I only show a few types of RNAs to make this clearer, but you can see that in a few samples really stand out here, because they stand out simply because there's much more microRNA present in the RNA. So the percentage of reads mapping to microRNA shown here in red is significantly higher to the others. And those reproducibly for the 4 pools that were processed were always these EV depleted supernatants. So, everything where EVs have been centrifuged, it's just protein bound RNA essentially. So that already gave us a first indication that probably there's more microRNA here associated to the protein complexes rather than the EVs. And this is also what you can see here in this heat map that is from the spiking normalized copy numbers. So you can see here a few clusters, essentially 3 clusters. You have here this group. This is the protein-bound microRNA, so outside EVs or the EV supernatant from the platelet release sets. This is from PPP. It's right here. It's from plasma. So this is microRNAs in plasma associated to protein complexes. Then we have here the platelet release set clustering, and you can see here that compared to plasma, there is actually a pretty strong and large cluster of microRNAs appearing at high concentration only after we have activated platelets, which in platelet poor plasma is not very abundant. And this is actually where we potentially could find good microRNA biomarkers of platelet activation. And it's certainly not in the EV, because small and large EVs, you can see here, there's pretty much absence or low levels for most microRNAs. So that's quite interesting for us. So when we specifically look into platelet activation, it's more in the protein fraction where we have to look at. So the conclusion that we took here is that in plasma, microRNAs are mainly localized in protein fraction, and only a subset of microRNAs is present in extracellular vesicles. platelets, according to the data that we have generated last, mainly releases microRNAs in association to proteins, which was a bit surprising for us. But I have to say that I'm not quite sure how generalizable this is, especially also to other biofluids. So I think for these kind of observations and studies, we have to be very transparent on what type of sample we do this, and you've seen that serum versus plasma can already make a difference. Also, you know, the EV field is still debating what are the quote-unquote right methods. I think there's no right method. You just have to understand what your method is doing. So these observations and conclusions may highly depend on the specific details of an experimental setup, but at least try to show you some of our insights into the potential utility for extracellular vesicles for microRNA or small RNA analysis. And now in the last 5 or 10 minutes, I'd like to now walk the bridge over to sort of the long RNAs and show you a few insights that we have made there. And I think that's quite exciting, because when you look at databases such as the xRNA Atlas, you know, where people, these consortia try to categorize data on RNAs in different biofluids, is still a strong bias towards, you know, microRNAs. And there's relatively little on especially cDNA, mRNA, etc. And simply it's because people have mainly been using small RNA sequencing. And small RNA sequencing has one, well, not problem, but one fact: it requires a 5' phosphate on your target RNA so that it's sufficiently or efficiently ligated in your sequencing library. Yeah, so you may have an underrepresentation of mRNA-derived fragments or long non-coding RNA fragments, where we assume that when you look at size distributions that they are fragmented, yeah, or even circularized RNAs which simply don't have this, yeah, because of the lack of a 5' phosphate. To address this, there are actually two publications, one from the Tushel lab and one from Munish Tevari, appearing in 2019. Back to back, they published on a very simple treatment of RNA, which is called PNK, or polynucleotide kinase treatment, which essentially the concept is that your long RNA fragment that has a 5' hydroxy and is and a 3' phosphate is converted into a fragment that suddenly has a 5' phosphate and a 3' hydroxy group. So it's more amenable or to be represented in a small RNA sequencing library, as it now resembles the features that are required for the ligation protocols. And what they observed really is, when they compared for cell-free small RNA from serum, EDTA, plasma, etc., untreated RNA or untreated small RNA, they saw a fairly high percentage of reads mapping to microRNAs here in orange, only a small fraction mapping to mRNA in red, or ribosomal RNA in yellow, etc. And after they had PNK treated the RNA, to achieve the 5' phosphate, they have seen, first of all, a strong reduction in microRNAs. You can see the red part goes down, and it's sort of replaced, I would say, by mRNA to a certain extent, but mostly by ribosomal RNA. You can see here that the yellow part takes over. Yeah? So it does have an impact, and they did some further analysis, and they have been able to show that they get more or better data on some of the longer RNAs, especially ribosomal RNA, but mRNA from that. So that's one option. And the second one, that a little bit later, again, two publications coming out on actually using not a small RNA sequencing workflow, but highly sensitive whole transcriptome sequencing workflows. And one of these is from Takara Bio. We're also using this. Again, we have no relationship with them. It's just something I'd like to share with you. We're using the SmartLess system, which in the version 3 has been optimized for really very low RNA inputs. Yeah? So this is a classical whole transcriptome sequencing workflow that also has the advantage of ribosomal RNA removal, of unique molecular indexes, and UDIs, so high multiplexing. Yeah? So something that actually also from not all small RNA sequencing workflow do utilize UMIs. Yeah? So that's an advantage. And what we have been interested in then in this study, to just understand the performance of both. So we had plasma from one aliquot of the plasma pools, we isolated extracellular vesicles. From the other, we directly isolated extracellular RNA. So we had plasma extracellular RNA and plasma EV extracellular RNA, and both we analyzed with the PNK, or this phospho small RNA sequencing workflow, and with the whole transcriptome sequencing workflow called SMART. And we try to understand, how well do the data sets compare. So first of all, when we look at plasma extracellular RNA, so directly without EV enrichment prior to RNA isolation, we do see that, as expected, small RNA sequencing just gives us read lengths of shorter size. You have actually a peak at a fairly low sequence length, and it extends maybe to around 50 nucleotides. However, with the whole transcriptome sequencing, we always got to 100 nucleotides or base pair sequence length in this, which is an advantage, because when you like to annotate or map mRNA, it's always good to have longer read length, because you have less problematic mappings that map to too many genomic loci, so have more unique mappings, and it allows you a better utilization of your data. And that's what we've seen. So we then compared in terms of cDNA and other non-coding RNA reads. We've compared our output from phospho-small RNA-seq, and we got around 14,000 different transcripts that we could map from the phospho-small RNA-seq data. But when we then compared this, and again, this is not just mRNA, it's cDNA and other types of non-coding RNAs, with the SMART or the whole transcriptome sequencing, it adds a lot more information. So there's a fairly large overlap. So SMART that detects pretty much, or a very large fraction of what we can detect with phospho-small RNA-seq, and I think this simply may also be some artifacts or sequencing depth. So I would say more or less it includes what you can get with the modified small RNA-seq, but it has much more information to it. So this is shown that in terms of the complexity, it seemed to be really the preferable workflow to use whole transcriptome sequencing. And then clearly the next question is, what can we potentially gain or lose, similar to what I've shown before on the microRNAs, by doing the EV enrichment. And so this is only first data, but we were quite surprised to see, in contrast to microRNAs, where there is a clear separation into those EV enriched and protein enriched, we've seen a very large overlap. So only focusing on mRNA now, or cDNA as a reference for mapping, we've seen that most of the transcripts we can detect actually in both, and there's only a relatively small set that's exclusively either present in EVs or total RNA isolated from plasma. And when you look at when you map these reads, and you try to understand the reads that we find in these data sets, in these whole transcriptome data sets, and we try to understand, a reference gene, beta-actin, for instance, how you have different transcripts of beta-actin, and how do these reads obtained from the circulation from the plasma EVs or the plasma xRNA map. You can see when you look left and right, it's actually very comparable. So this also suggests that, first of all, the alignments really suggest that this one is the main transcript that we do find represented in the circulation. And when you look at the pattern of these peaks, it's very, very similar. And overall, the performance when we do this on replicates of plasma xRNA and plasma EV xRNA sequencing works really well. So these are also, I would say, not more or less random results, but this is also highly reproducible. And so this is my final conclusion. And I find this is really only the starting point for us to look into long RNAs using whole transcriptome sequencing. But what especially the last result tells me is that there seems to be the much higher importance of extracellular vesicles, because there's a much stronger overlap, or predominantly one could say, maybe a bit exaggerating, that the majority of mRNA that we are detecting may be associated to extracellular vesicles in the circulation. While we have seen with microRNAs, this may be the opposite. So, we're working with EVs, this may actually be quite an interesting readout to apply whole transcriptome sequencing and look into long RNAs. So suggests that cell-free mRNA may be predominantly or mainly present inside EVs. So I hope that with this webinar and with some unpublished data from my lab, I could show you in these 4 areas a few new results, maybe give you something to think about, maybe something to adopt for your own research. Again, to summarize, the concept of why absolute normalization may be a good practice or a good add-on for small RNA sequencing. Again, if you're interested in this, just let me know. That the enrichment of quote-unquote active, so risk-contained microRNAs, especially from cells and tissues, could really give new insights into importance of microRNAs or identification of biomarkers, but also mode of actions. And combine the last part, what can we obtain from extracellular vesicles? And at least we've seen for microRNAs, our observation is that in plasma, at least, microRNAs are predominantly outside EVs, more in protein complexes. But when we move on to longer cell-free RNA, this may actually be the opposite. We'll see. Yeah. With that, I just would like to acknowledge my team at Tamerna and alumni and our back office and partners. So we're a fairly big group by now. If you're ever in Vienna, just let me know, and I'll be happy to have you and have an RNA discussion. And then finally, also of course our funding agencies, and of course also Miron for inviting me here to speak to you today. And hopefully we'll have some time for questions or in a discussion. So thank you.
Thank you so much, Matthias, for this. I'm very pleased to see that Tamerna team really grow over the past couple of years. I remember that we first talked in 2000, I think maybe 19, early 20, when we met together, and now I see that the team is much bigger, which really pleased me. Thank you so much for these insights. I think that all the people who are really into RNA have can't wait to ask questions, but also for other people who are maybe not really into RNA that much, but has a good now get good insights about which types of things we need to really be focused when we are going into this kind of biomarker approach, at least when it comes to RNA. I have a tons of questions here, but I will be a good host and let other ask you. First, we have one question in the chat, and then Fabio will join us to ask the question. Just let me see. I think Karine asked, what kind of bioinformatics algorithm are you use for identification of microRNA biomarkers? So maybe it's a little bit more general question, but hopefully we can.
Yeah, so we actually published this, so you can read everything up in the paper. 2022, when you Google for MIND, M-I-N-D, it's there. And I'm not a bioinformatician, but high level, we are using MIRDeep2 for, so after, processing the raw data and a bit of cleaning for the annotation and mapping is MIRDeep2. And then for, basically doing differential expression, we use DSEC2 and HR. This is sort of, basic published packages, and we've integrated this in a pipeline that we call MIND. That's for the basic part, and then we have clearly for annotating the MIND spike-ins that we are now using those a bit of additional annotational mapping going on. Yeah, and then to then use this data and maybe develop signatures or multivariate models. Then we don't have our own. I mean, we're really agnostic. We're using different concepts of AI and modeling, starting from multiple logistic regression really to more decision tree based models, support vector machines. So, and there's really not, I would say it really also has, the method has to fit your data, the size of your data, how many replicates you have, or how big your sample sizes is, your strategy for. So I think it's, unfortunately, there's no generalized answer on how, what best approaches are then for this kind of signature or multivariate biomarker development.
Okay, thank you. Let's try to unmute Fabio. Fabio, I sent you the request to unmute, so it should be okay now.
Can you hear me now?
Yes, yes.
Okay. So, hi, Matthias, great talk. About last part of your presentation, so looking at long RNAs. My question is whether or not you try to measure whether circular RNAs are also in your among your reads, and whether or not they have similar distribution as the linear RNAs. Theoretically, circRNAs should be promising candidates as biomarkers, being less sensitive to exonucleases. So
Yes.
if you looked into it.
Yes, we've looked into it. We are still new, also from a bioinformatic point of view. You know, it's quite challenging. We've used a few published tools, circRNA Explorer, for instance, but it's a bit premature to really present it. That's why, you know, I'm a bit hesitant, and, you know, happy to also get some feedback from, you know, some like you guys. You know, you have a lot of experience in the field. But yes, we've started to look into that, and we can recover circRNAs, at least with the bioinformatic tools we've been using. We haven't really explored yet, you know, redistribution between EVs, total RNA, you know, whether it follows the same trend as for the mRNA as I've shown you, but that's something that we'd like to understand. Yeah.
Yeah, thank you. And if I may, a second one, just this relates to the first part of your talk. So I like your concept of absolute quantification with a normalization with the spike-ins. But we also have another element that is important, that we receive samples from the clinic, and our clinicians are often very busy and sometimes don't really respect the guidelines we give them. So since these spike-ins are added at the moment of extraction or after extraction, so is there a way also to check degradation of the sample that may have occurred before enters the lab? Generally, when it comes to the lab, everything is less complicated, let's say.
Yeah, it's a very good question, similar to the first one. So yes, for us there is a way, because when we internally, when we process, you know, some serum plasma that's coming from a clinical source, we standardize and we use for the RNA extraction defined volume. You know, 200 microliter somehow, I think, has established itself. We use 200 microliter, and then what we do is from this. It's a standardized workflow, and we elute the RNA from this 200 microliter plasma into a defined volume of 30 microliter total RNA. And then from this, we take again a defined volume, I think it's 8 microliter, and add 1 microliter spike-ins. So we don't really mix the spike-ins to, at least for biofluids, to measure concentration of RNA, but to a volume. Yeah? And if in that volume I have complete RNA degradation, because whatever was happening before that sample got to us resulted in a very low presence, we'll see this in the sequencing. Then we have a spiking calibrator, but we see that all of the reads for our RNAs are in the, you know, in the low range. Yeah? And that has happened to us with, for instance, also some EV samples and so on. So yes, if there's an overall degradation, you can simply see that you know, the distribution of your microRNA spike-ins relative to of the microRNAs relative to the spike-ins looks just different. Okay.
So I mean, somehow reading the number of reads still has its importance. The number of reads that you get as opposed to the numbers of reads that you get from the spike-ins. It's the ratio that you look at, essentially.
Exactly, it's the ratio. Yeah. Mm-hmm.
Okay, thank you. Do we have any other questions from the audience?
I just have a comment. First of all, if I may, really an illuminating talk, because really you are telling us that, I mean, the pre-analytical phase is not complex, is horribly complex. Which means that first, when you read papers regarding transcriptomics, I mean, what do you actually read? What are you actually reading? Am I exaggerating?
Yeah, that's a good point, because.
And then the other point is that we may be careful. We may start with platelet poor plasma, but then EVs, and then EVs of which size, which EVs. So I mean, we really need to kind of find probably to standardize somehow some procedures that can just be platelet pool. I'm not an expert in the field. I'm just stepped in thanks to Miron and Dimitris and other friends. So it's very important. So the knowledge you transfer to us should be transferred, maybe simplified to everybody collecting samples, because this is something absolutely, I mean, the relevance of what you really obtain. I agree with Fabio. I mean, you cannot just get whatever blood is there. Sorry. So I'm not simplifying the things. I'm just saying that you illuminated us, many of us, into the complexity. And then, of course, if you want to have not just a paper published, but data that are actually promoting, knowledge, we need to take into consideration all these things. Not only, of course, in your company, because you're working in a wonderful way, but really everywhere. So this is just a general comment. Of course, normalization, absolute values. I mean, these are all things that we need to learn to take into proper consideration. So,
yeah, no, and absolutely, yeah, and for us, I didn't talk about our diagnostic test, but just to give you an example, our diagnostic test has twice as many quality controls on the assay than it has biomarker targets. Yeah, because when we report the result, ultimately, that really will be used to decide on, whether to operate or not on these liver cancer patients. We have to make sure, and so we monitor for hemolysis, for inhibition in the sample, etc., etc. Yeah, and that's the learning that's one of the main learnings that I also have made, that it's that everything that happens before you generate data can really have a major impact. Yeah. And I'm not sure this is only true. I mean, I'm an RNA person. I couldn't comment on proteins, but I would be surprised if this were not, or metabolites or other classes of biomarkers as well. So I think the general awareness of the importance of this is just very important.
Yeah, I think that's something that's really, when you see the pipeline of research, the only thing that you cannot really go back is take the sample from the patient at the point when it was collected. Everything, and ironically, all the other stages of sample treatment we control. What Fabio mentioned, when it comes to laboratory, it's much easier to control things, but that trip from the patient to the, while it is in your hands, sometimes it can be very spiral and it's really hard to control the things that are happening between the patient admission, I would say, not just the sampling, but the admission and the things that we are doing in the lab. I would also kind of comment and ask you to give a more explanation on these technical details. It is also for my sake, but I think for audience sake. So you mentioned several times different types of samples from blood. So let's just focus on blood as a major biological specimen that we use for biomarker discovery. We know that there is a whole blood, there is PBMCs, there is PAX gene, there is platelet poor plasma, there is whatever it is, and whatever people define platelet poor plasma in papers, you can find different definitions. We have serum. So what would be your kind of preference if you go into biomarker discovery? I guess it may depend on the pathology and on the hypothesis, but how would you approach your decision what type of blood derivative you would use for biomarker?
Yeah, understood. Yeah, it's a good question, and I've also learned about this. So first of all, even taking a step, it doesn't always have to be blood. So our approach is now when we go in maybe in an orthopedic disease, maybe a joint disease, or in a kidney disease, is there maybe another biofluid that's closer to the organ or the cell system? So kidney disease is urine, clearly, maybe really suitable because there's maybe more material shed there, and you have, again, signal to noise. What I said initially, you may have higher signal in a biofluid that's close to the cells or the organs that you're interested in. Yeah, so for instance, I can tell that we're working with synovial fluid now for specific diseases affecting joints because it's reasonably non-invasive or minimal invasive, but it's just very close to the site of disease. Yeah, so your signal to noise is much better. So I would always consider this when you have a certain disease or organ were you interested in a biomarker? Is there maybe another, because in blood, ultimately everything ends up there, you know, and we've seen that when we try to then bridge maybe from synovial fluid to plasma, it's challenging because your signal to noise ratio is smaller. There's much more competition of your biomarker with maybe that same biomarker also secreted than from other organs. Yeah, unless you have something that's very, very cell type specific, then you may have the luxury to always go to blood. Yeah, but if you're let's talk about blood, and there clearly, I mean, if you use whole blood, maybe PAX gene tubes, you're measuring mainly signal coming from blood cells, erythrocytes, platelets, the cell-free and the RNA that may have been shed from other organs is really underrepresented in the whole blood sample. So, but that doesn't have to be bad. I mean, if you're looking into inflammatory diseases where you do expect a lot of signal taking place in different blood cell types, you know, it may actually be good, and it has certain advantages. Yeah. Platelet poor, platelet free plasma is now for most microRNA work, and we're still doing, I would say 90% of our work is on microRNAs. We always try to convince people using plasma and then platelet poor, platelet free plasma, because there's less interference from platelets or blood cell types in this, or less background. We're trying to, but sometimes you don't have the choice. And then again, something I wanted to say before on pre-analytical, the other what we have to be careful is that these generalizations that we think, okay, microRNAs are sensitive towards hemolysis, is also not true, because we have 2,500 microRNAs, each microRNA having its own promoter, its own distribution across cell types, and we have a lot of microRNAs, actually 20%, I didn't touch upon this, which are so-called tissue enriched or cell type enriched. MicroRNA 122 in the liver is a good example. If you measure microRNA 122 in the circulation, results between serum and plasma are highly correlated. The reason is it only comes from hepatocytes. There's no interference of hemolysis, so it's a really nice biomarker. So we also have to be careful that we don't generalize. We just have to treat each microRNA really as its own biomarker and understand, you know, what are these interferences, what's the best matrix, similar as we do with proteins. Yeah. Also with proteins, we don't say proteins in general behave like this or that. There's some that you have to be more careful with, and some that are quite easy. And I think the same probably applies to RNA itself. We just have to find out and test.
Thank you. When it comes to platelets and the paper that you published about the microRNA in platelets, so from my perspective, when we do kind of preparation for platelet poor plasma and we centrifuge in 10,000 Gs in order to obtain it, we have a very small or non-visible pellet of platelets there. So what is your starting volume when you want to study platelets? For example, if we would like to study platelets in the future and microRNA in platelets, from which volume we should start?
It's a good question. You caught me a bit off guard because I never actually did it. It's my team who does it, but I do think that, right? I mean, from the, at least in the first centrifugation step, even from 1 or 2 mLs, you should have a fairly good amount of. But it's a good point. I'd have to go back to the team actually. I'm sorry, I couldn't really give you a reliable answer to that question.
So starting about 1 milliliter should be more or less okay for obtaining that. I would just end up with 2 more things, and then for me at least it's done. One is also, you touched a little bit this about the exosome and lipoproteins. We know, I mean, I know that in past 10 years there were some reports that HDL, LDL particles, which are we as people that are focused on cardiovascular disease very obsessed about. There was some data that certain microRNAs are also carried by these. On the other hand, when you look how these molecules, how HDL and LDL was separated in these methods, and then when you look the physical chemical properties of HDL and exosomes, you can see that their density is quite similar. So I was always wondering, can you be relatively sure if these are microRNAs coming from lipoprotein particles or from exosomes, or is there any cross? I mean, I'm not big expert. I'm not expert at all in exosomes. So I'm just wondering, when you go to exosomes, how sure you can be it's only exosomes, or if you go to lipoproteins, how sure can you be it's not contaminated by exosomes?
Yeah, it's a good point. I think to really tell that takes a lot of analytics of your sample, to really be sure what to characterize what you have, which is mostly not done. So when you look in literature, I think that's what I tried to also in my conclusions to say, okay, we make certain assumptions, but there are a lot of factors to this, changes in protocols, different EV isolation, SEC to ultracentrifugation to densing gradient, and that may change these observations that I've shown today. Yeah, so we have to be really, I think what's most important in that area of research, to be very transparent with what has been done, so we can really interpret the results that we see in papers. Yeah, be very transparent about the methods that have been used. And also to acknowledge that we just don't know whether the preparation ultimately that we've analyzed is actually really truly exosomes or may have lipoproteins still in it. Yeah, so I think that's certainly a caveat to all of that, and I think we can still learn much more about this.
Okay, and one last point, which is kind of for people that are just entering the field and want to start discovering microRNAs and so on. I mean, you and me privately discussed this many times. It's about, in the discovery phase, there is always a question: what is the effect size? Is there some normative to that? And also on the other side, what should be the sequencing depth? I guess it all depends on the level of expression and so on. So I would like to kind of have from your side, from your expertise and experience, the kind of definition or a range of sequencing depth. For example, in case we just want to do mRNAs and differential expression, and then if we want to use microRNAs, and then if we want to go to long non-coding RNAs. I know that always sequencing depth should be more and more, but if you can just shed some light on these topics.
Yeah, definitely. I mean, I was a bit fast. I can actually go back and I don't know if I can share my screen.
Yeah.
Because I think it's let's see, share this one. Yeah, so that is this slide. It actually shows the sequencing depth. So read counts total is more or less sequencing depth in millions. So this is 20 million, 40. And you can see that most of these, at least microRNA or small RNA sequencing we do on biofluids is in the range below 20 million per sample. But actually when we design it, I mean, sometimes, you have variability and you pool and you multiplex with a certain idea in mind, but sometimes you add up a bit more, but actually even more is below 10 million. And with 10 million, I have the feeling that we can really exploit most of, or have a sensitivity where we see most of the microRNAs that we can detect, because you can see that here, even if we sequence with, and this is plasma, we have most data. If you go to 40 or 60, you don't get at least with our assay, we don't get 2000. We level off maybe at 1200. But these are low abundant. So the question is always, when I look for biomarkers, I do try to get something that has a fairly good measurability. So long story short, in terms of microRNA, small RNA sequencing, we aim for 10 million, 7 to 10 million for plasma. And for tissues, even less, because in tissues, we usually have a fairly high percentage of reads mapping to microRNAs with our protocol. So maybe there's 5 to 7 million. That's all we need for exosomes. Since exosomes or EVs have a fairly low abundance, we try to get a bit deeper. I have to say that. That's for small RNA sequencing. For the whole transcriptome sequencing I've shown in the last part, we try to go fairly deep. There we definitely want to go maybe 30, 40 million. Because we've seen that, first of all, you have a higher complexity and we don't have that much experience that we could generate plots like this from the whole transcriptome sequencing of cell-free circulating long RNA, but my impression was that we have to sequence deeper. And then it also, for cells and tissues, it also depends a bit on the sequencing workflow that you're using. There are some, 3' sequencing, quant-seq, where you can actually get away with very as they are very targeted to your to the poly A and to the 3' end, you can get away with even lower sequencing depth, 3 to 5 million. Yeah, but if you don't want to do whole transcriptome sequencing, where even circ RNAs, you know, Fabio mentioned circ RNAs, which still you have to get these reads that span these backsplice junctions, you know. I mean, Fabio can tell you, I've seen a poster where I think they go 50 million, 100 million reads. Yeah, so I think there is unfortunately not no one fits all answer, and it's again depends on a few things there. Yeah.
So this is all for biofluids, and for example, for tissues, for like classical differential expression between, that's what would be your kind of...
Yeah, for tissues, when we classical gene expression analysis, differential expression on mRNA level, we call this, we have the quant-seq workflow. It's a 3' end sequencing. Then we actually just go to 3 million reads. Yeah, that's fine. But we could not do from that data, you know, looking at different isoforms, etc. It's really just measuring gene expression and trying to understand it. Yeah, then you can get away with much lower. Yeah.
Okay. Now, Fabio just confirmed 100 million reads.
Yeah, 100 million reads,
for circ RNAs.
Yeah, it's expensive. Yeah.
Okay, that's for me. I don't know if anybody else has any other questions. If not, then I would like to thank Matthias one more time for his dedication and time, and politely giving us very nice insights into methodological complications, I would say. Thank you so much. Thanks to everybody else, and see you soon. This was recorded, so it's going to be published on our YouTube channel of both projects. So anybody who is interested can also go through this lecture and get the information again. Thank you so much one more time. See you soon. Sometime in March, we will schedule next webinar, but you will be all notified about this. So thank you so much. Have a nice week and nice weekend, and see you soon.
You too.
Bye bye.
Thank you all.
Thank you very much.
Have a good evening.
For attending. Bye bye.
Bye now.